Sorry for the delay there. So it's a real pleasure to introduce Satchin Panda. Satchin got his bachelor's and master's degrees in India, and then he came to the Scripps Institute in San Diego to do a PhD with Steve Kay, and he got his PhD in 2001. So this was the time of the, I would call the golden age of mammalian circadian rhythm research. So he then stayed in San Diego and went to the Novartis Institute to do a postdoc with John Hoganish. And he became an assistant professor at Salk in 2004. So, you know, the circadian system is a global regulator of pretty much everything. So, you know, there's that old saying, think global, act local. And Satchin's work is really reflects that philosophy, as you'll partly hear, looking at specific cell tissue type effects of circadian disruptions. And, you know, nowadays we know that the circadian system even has subcellular ramifications, has effects on all sorts of cell biology processes, and so it's even more local. And in the face of that, I think there's this question like, well, we know diseases, we know aging, we know everything under the sun correlates with circadian defects. And in fact, some circadian defects can cause defined diseases. So how do you act on that? How do you act on something that's so global, yet can be so local? And Satchin's work on time-restricted feeding is, I think, the most encouraging avenue to pursue because we can, he's shown that you can do this in organisms ranging from model organisms up to humans.
Thank you for the awesome introduction. Actually, there's a lot of burden on my shoulder now to live up to that expectation. And it's actually a pleasure to visit University of Wisconsin at Madison, because when I was a grad student, I used to work on Arabidopsis, and then the Arabidopsis genetics conference used to rotate and used to happen here. So I visited the campus many times during my PhD. The last time was actually in 2001. So it's nice to come back after 22 years. So let's see. Okay, so before I start, I would like to thank lab members who contributed to stuff that I'm going to talk about, and on the other side are the funders who have been funding the work, and there's my disclosure. Okay, so I guess if we think about what is the goal of biomedical research, it is to enable everyone, irrespective of age, gender, ethnicity, and health condition, to live at their peak performance, that is physical, emotional, and intellectual performance at every age of their life, because living at our peak performance is a fundamental human aspiration and also human right. And along that line, if we think about health, most of the cases, we want to treat disease, because there are many age-related disease, there are many gene-related genetic defect of disease. But what is interesting is for every person they do health, the 10 highest grossing drugs in the United States fail to improve the conditions between 3 and 24 people. So that means the best drug that you can bring to market, even if it treats 1 out of 25 people, you are moving the needle. That's the positive side. And then the depressing side is, or the hopeful side is, there is a lot of headspace to improve human health. And when you think about why some of these drugs don't benefit, the first thing that comes to mind is maybe the drugs do not match with lifestyle. For example, if you give a drug, blood pressure lowering drug, to a person who continues to eat high salt diet, you don't expect too much benefit. So that's why what we think is two things. Lifestyle can prevent or delay the onset of disease, because this picture is telling us that it's not good. You should not be very hopeful to have a disease, because the success rate is pretty low. You can prevent. And second is, once we have the disease, can we combine lifestyle with disease to improve prognosis? So then the question is, what is lifestyle? Because lifestyle is a very fuzzy term. It can go anywhere from Mediterranean diet, high protein, low protein, vegan diet, creatine, all this stuff in between. So the way we look at lifestyle is what, when, and how much we eat, sleep, and move every day. And lifestyle is something that we don't do as an intervention. So for example, being on ketogenic diet for 3 months is not lifestyle. It's an intervention. But what we do on a daily basis, that's what is lifestyle. And we don't discount that what and how much we eat, sleep, or move are not important. Those are important. And in that context, quality and quantity of nutrition, for example, are important. But at the same time, when or the timing is also sometimes, in some cases, maybe a dominant determinant of health. So that also relates to the concept of circadian rhythm or 24 hours clock. And the bottom line is, what we know now is our circadian rhythms are regulated by cell autonomous circadian clock that are present in almost every organ, including our brain, and particularly the hypothalamic SCN, suprachiasmatic nucleus, harbors the master circadian clock, and that sends diffusible signals or systemic signals directly or indirectly to the rest of the organs that have their own clock to be in sync. And that leads to daily rhythms in physiology, metabolism, behavior, and even gut microbiome. And as we evolved on this planet, where the sunrise to sunrise is not exactly 24 hours every single day because of the tilt of the planet and the change of the season, the clock has to be entrained to the outside world, and that usually happens primarily through light perceived through the eyes. And that's how we have been designed to adapt to environment. And over the last 30 years, the progress in molecular circadian rhythm research can be summarized in this cartoon, where clock and BMAL1, which is also known as MOF3, member of fast domain containing protein 3, heterodimerize and bind to E boxes present in front of cryptochrome and period genes in mammals. And that in turn act as repressors of clock and BMAL that produces rhythms in cryptochrome and period genes. And then clock and BMAL also drive the transcription of ROR and reverb class of nuclear hormone receptors, which in turn act as activators and repressor of clock and particularly BMAL. And this interlocked feedback loops can drive also many other genes, because these are essentially transcription factors. And in this context, I must highlight that the initial MOF3 or BMAL1 binding to circadian factor was actually discovered in this campus by Chris Bradfield when John Hoganes was his PhD student. And when I was a postdoc in John Hoganes' lab, that time Trey Chateau, who is actually here, who is also in this campus, he and I were the only postdocs in the lab. Actually, yesterday we had our 20 plus years reunion of the lab, so all the postdocs, so there are 2 of us. And ROR was established as one of the core clock component of mammalian circadian system. And around that time, what was interesting was until 2000, 2001, it was also known that many, not all, blind people who have lost their rod and cones can still entrain their circadian rhythm to light-dark cycle. So in John's lab, one of my side projects was to figure out whether this melanopsin is in fact one of the missing photoreceptors. So the reason why I'm bringing this up, a lot of people ask what is the relative importance of light and darkness versus feeding and fasting in circadian rhythm. So we established that melanopsin is required for normal light-induced circadian phase shifting. And then subsequently, we also showed that melanopsin and rod cone photoreceptors account for all the ocular photoresponses in mice. And over the years, we have been working on melanopsin. And recently, we have focused primarily on human retina because we realized that although melanopsin was discovered and described by the turn of the century until 2019, 2018, there was no direct recording of melanopsin function from human retina. And over the last few years, we have been collaborating with Life Sharing, an organ donation program in San Diego, and an ophthalmologist, Ann Henneken, to have access to human retina within, right now, we can go down to 20 minutes after death, and we can revive almost all visual photoreceptor function in human postmortem retina. So this is becoming an exciting area of research in my lab. So how do all of these 20 years of research relate to human health? Bottom line is melanopsin is a blue light sensing protein present in only 5,000 retinal ganglion cells in the eye. They're hardwired to the master clock in the brain in the sense that they're monosynaptically connected to master circadian oscillator neurons in the suprachiasmatic nucleus. Since they're less sensitive to overall light, our ancestors, when they had candlelight or moonlight, essentially after evening, melanopsin was not active at all. So that allowed the sleep hormone melatonin, which is strongly repressed by light, to rise at night, which resulted in better sleep. And during daytime, melanopsin needs a huge amount of daylight to be activated. For example, even standing here, I'm getting almost 250 lux of light on my eyes, and most of you who are sitting there are getting 50 lux max, and that's not enough to activate melanopsin. You have to be outdoor. Even on a cloudy day in Wisconsin, you get 5,000 to 10,000 lux of light outdoor, and that's pretty good in activating melanopsin. So it synchronizes brain clock, raises alertness, reduces depression, and in modern days, as we are stuck in throughout day and night, we are glaring at rectangular pieces of glowing objects. It activates melanopsin at night, and we don't get enough light during daytime, so that leads to sleep problems at nighttime, and then we depend on coffee in the morning. So essentially, our life these days goes from coffee in the morning to popping melatonin pill at night to address these issues. But over the last several years, people have in mouse model and from human studies that this light dark disruption or the insufficient light during daytime or too much light at night chronically can increase risk for many of these diseases that are listed here. So that led to the idea that lighting can be manipulated for health. And this is a big thing in lighting history because almost 100,000 years ago, humans controlled fire. And for the first 100,000 years, or minus 150 years, light was mostly for warmth, for cooking food, and to light up the evening for 2 or 3 hours. Over the last 150 years, during Industrial Revolution, lighting became cheaper. So lighting is also for safety and for evening activity, but we never thought light in terms of health. So right now there will be, and that is actually happening, it's happening now, what we call light therapeutics. So that means human-centric lighting is coming. In fact, 4 years ago, for the first time, the International Space Station was retrofitted with human-centric lighting to simulate blue enriched light during daytime when Houston is working, and then less blue at nighttime. And then the question is, how much light is healthy? What we now know from human circadian rhythm research and also from epidemiology of depression and seasonal affective disorders, to reduce all of these depression, seasonal affective disorders, or ADHD, 10,000 lux of light, or being outdoor for 15 to 60 minutes is pretty good. And an average person, for all of us, we should get 1,000 lux of light for 30 minutes or more, so which is equivalent to sitting even next to a large window. And at nighttime, there should be less than 20 lux of light in bedroom and your living room. And we should have at least this light management. Question is, well, these are common sense. Is there a huge impact? In fact, in the US, 1 out of 10 births now is preterm. That means these babies are born before the week of 36. 8% babies are born between 33 and 36 weeks of age. And these babies are put into NICU, neonatal ICUs, which are mostly in constant light. So there was a nice study done in Mexico City where they simulated light-dark cycle by putting a cover around baby's head, not to suffocate, but there was enough space. So essentially, during daytime, there are 250 lux of light, and during nighttime, there was 27 lux of light, and that led to accelerated growth and discharge of these kids from NICU by 13 days. 1 day of NICU care in the US costs somewhere between $12,000 to $17,000. And we're talking about 13 days of reduced NICU, median NICU time. So just do the math. There are 300,000 premature babies in the US. If we just implement this, there is not even a single drug that can accelerate baby's growth in NICU setting. So this is way much powerful than any drug. And then just imagine if we implement similar light-dark simulation in any ICU, because 30% of ICU patients go through delirium, and that can be prevented if we simulate light-dark. So that's one end of the spectrum. The other end is, let's see. Oh, this ran out of battery. In collaboration with Horacio de la Iglesia, we have been also monitoring when do people used to go to sleep, how much light they were exposed to. So this is an Argentinian toba who have no access to electricity, so they live under candlelight or firelight. So this is the time when they get the last 5 lux of light, that's the bottom, or the first 5 lux of light. 5 lux of light is the amount of light that's at your feet under the chair. So that's the amount of light, that's 5 lux. So that means when these people are getting up and getting out of their hut, just before getting out of the hut, they're seeing that amount of light. And in the evening, you can see that as soon as the sun goes down, they don't have any exposure to bright light. And we always thought, okay, so what time our ancestors used to go to bed? They actually used to go to bed between 9:30 and 10 p.m. because they light a fire and they cook and they share a meal. And the fireside chat or fireside social activity is the cradle of civilization, so they do all of that. So now, if we measure with the same instrument, Seattle high school student, and the band there that you see is 45 days of data, so that's the standard deviation. And so that's a Seattle high school student, one student, and that's another student. And if you look at UCSD students, they're not different. So essentially, in modern days, our kids, particularly teenagers, we don't know why, there seems to be teenagers are more sensitive to evening light than adults. So it makes sense because we know that in the same household, the parents should feel sleepy around 10 or 11, but the teenage kids, they can stay up till midnight or 1 o'clock. So there is this developmental or hormonal change in light sensitivity. We don't understand that. But the bottom line is they are programmed to go to bed late and also wake up late. And another thing that we find is among Argentinian tobas, this is the physical activity. What is interesting is we have been designed to be physically more active in late afternoon because that's when the tobas are running back from the field to home, or people who were living at home, they're kind of trying to get everything together, bring everything indoor because it's scary outside. The bottom is activity from high school students, 100 high school students. Each cohort has 100. And what you see is there are tiny spikes throughout the day. That's the time high school students go from one classroom to another classroom. And you'll see the same thing here in Wisconsin. And this was done in 2 different high schools. One was a rich neighborhood, one was a poor neighborhood, very similar. There is no difference in activity in weekday and weekend in tobas, but of course, as you'd expect, the kids you can see that in the weekend they're sleeping late and they're also less active. So that led to the idea that yes, teenagers in the US are sleep deprived, and one way you can change or improve sleep is by delaying school start time. So in these two schools, Horacio convinced Seattle School District to delay the high school start time by an hour. And by doing that, we increased sleep time by 34 minutes, and that also correlated with 4.5% increase in the median grades. So that led to, this was one of the original studies, and then this has been reproduced now in Europe and in many places in the US. We also replicated it in La Jolla in San Diego. So the bottom line is now in California, all middle schools and high schools cannot start before 8:30 a.m. So this is one case where circadian rhythm science has led to public policy changes, which is equivalent to putting these kids on 2 sleeping pills, because 1 sleeping pill increases sleep by 15 minutes. And you won't do that to your kids, but public policy can do that. Okay, so I'll go back to, again, this slide. And I told you that many of these components are transcriptional regulators. So almost 20, when I was a postdoc, that time, John Hoggeness, I was in GNF. So we said, let's do a very simple experiment. Let's put mice on ad libitum condition, ad libitum feeding of normal chow, and then harvest liver sample and a CN sample in every 4 hours for 24 or 48 hours, and then see what is rhythmic. And what we found was, yes, nearly 300 genes in the SCN, 300 genes in the liver, they were cycling. And there are 6,000 probe sets on this AFI-RS, those old days. And that was very gratifying that we showed that circadian rhythm is tissue specific, because it's not the same 300 genes that cycle in the SCN or liver. It's a very different set. And as I started at Salk and John started in UPenn, we had our startup fund. So we said, okay, so let's go back, redo this experiment with better AFI-RS that covers the whole genome, and increase the sampling frequency to every 1 hour. And we did this for 48 hours. Food was available ad libitum, young male mice. What we found was, we found this 24 hours period that was expected, nearly 3,000 genes, roughly 2,900 plus or minus few, were rhythmic with 24 hours rhythm. At the same time, John also found and got super excited about this 12 hours period genes or transcripts that are cycling with 12 hours period. So that's why the title of the manuscript was Harmonics of Circadian Gene Transcription in Mammals, because what he came up with the idea was, maybe there is a clock in the brain that's in a different phase and clock in the liver in a different phase. They are interacting and they're producing this 12 hours rhythm. And that sounded very interesting. And at the same time, Willis Sibler's lab also published this paper showing that if you take out the liver clock completely, the mice are, of course, at that time, people are thinking about ad libitum feeding or feeding pattern, then you'd see some genes cycling. And his data essentially supported this idea that maybe the SCN clock somehow drives rhythmic transcription of some liver genes. And John was thinking, yes, liver clock and SCN clock can crosstalk and you get this 12 hours rhythms. When I looked at all those genes that are cycling with 12 hours and looked up the history of those genes, many of them seem to be food induced. So I thought, okay, so let's go back and ask, when do mice eat? And actually, male C57B6 mice, not female mice, they actually have a big breakfast and then they go take a nap and they come back and eat again. And the RER is reflected in that way. So we said, okay, if you feed mice day phase, so now mice are fed in the daytime, the SCN is tied to light dark cycle, then we should see more of these 12 hours rhythms. Or if it is entirely driven by feeding, then we should see only 24 hours rhythms. And the answer was, we saw only 24 hours rhythms, all the 12 hours rhythms disappeared. So that means most of these rhythms in the liver are driven by feeding and fasting. We took it one step further. If we take wild type mice, we don't give any food at all, then nearly 368 or give or take 50 genes 400 genes would cycle. So this is without any feeding cue. So we think that this is mostly driven by endogenous intrinsic circadian clock. Of course, as I told you, ad libitum feeding will lead to 3,000 genes cycling. And if we time feed only during daytime, those 3,000 plus another 1,000 genes, or 2,000 genes actually become rhythmic, and they all track food. So now the question is, are they driven, all the rhythms driven by food, or what is the contribution of genes? So if we take cry double knockout mice, they have no feeding rhythm. Under ad libitum condition, they will eat continuously. Then you get 20 genes cycling. That's the false discovery rate. You would expect 0.05% genes to be cycling. But if you consolidate feeding, then again, you see only very few genes, 617, not 5,000 genes cycle. So the bottom line take-home message is you need a functional circadian clock and feeding fasting rhythm to drive robust rhythm in transcription, at least in the liver. So until that time, most of the stuff that you knew about circadian rhythm came from mice. Mice are nocturnal, they have fragmented sleep, and they also have fragmented feeding time. So we thought, how do we know what actually cycles and when the cycle in humans? Of course, it's a very difficult experiment. You cannot do that experiment in humans, so we moved to baboons. So this was done in this experiment from concept to publication took 10 years. So we went to International Primate Research Institute in Nairobi, Kenya, where these baboons were captured and quarantined for 18 months. And baboons are actually considered pests in Africa. So they come and destroy crops. So the Kenyan wildlife goes and captures them, and then they sack them once in a while. So it's not an inhuman experiment. So we collected 64 different tissues, including 22 different brain regions every 2 hours, over 24 hours. And then what we found is almost every tissue has a set of genes that cycle. And if we combine this, what we found is nearly 82% of protein coding genes in baboons show a tissue-specific diurnal rhythm. So now you might ask, well, how are these baboons fed, housed, etc.? So these baboons are actually living in semi-natural conditions. So that means they were experiencing almost 10-degree cycle, a 10-degree centigrade cycle in temperature, because the ambient temperature in Nairobi, we measured that, it was cycling 10 degree. We had a temperature sensor on their back, and their core body temperature, not core body temperature, their skin temperature was actually fluctuating by almost 3 degrees centigrade. And this is the activity rest cycle. And they were in light dark cycle, and they were given 2 meals, just like in most primate facility. You may or may not know, most primate facilities, the monkeys are actually fed twice. In the morning when the vivarium staff come in, and then in the evening, either they feed or take away food. So that might explain why it's very difficult to produce a diet-induced obesity model in primates, because primates are given food only twice. And another thing that we found was there are 2 strong peaks in gene expression, one anticipating evening and one anticipating morning. And this was very different from what had been published in mice ad libitum fed condition. But it's gratifying to see that this kind of result is now reproduced in humans in many other settings. If we ask, what are these genes doing? If we do functional annotation of rhythmic transcripts, those are rhythmic in at least 20 different tissues, then what we find is many basic cellular processes seem to be diurnal, seem to have a diurnal rhythm. And then if you ask, what are these processes, and if they go, if any one or more of these are disrupted, then what happens? Actually, these are the basis for most of our chronic disease. So that's why it's not surprising that if we go back in literature and ask, what happens if you disrupt circadian rhythm by keeping people awake throughout the night, or disrupting their sleep, or now if you can feed them at the wrong time, then few days of disruption doesn't lead to too much of disease, but these are mostly discomfort. If we continue circadian disruption as in shift work, or also in mouse model of circadian disruption, then the risk for at least 100 different chronic disease go up. I'm not saying that it causes the disease, but the risk for the disease go up. And as a result, the circadian rhythm field, like if you look globally, what the field is trying to do to improve human health, we try to do three different things. One is to nurture better circadian rhythm, what we call training the circadian clock. And then second is clocking the drugs, because this is a new development in the field, that it seems like nearly 80% of FDA approved drug targets are also circadian. So that means if you match drug timing with the right, the timing of the target or timing of cytochrome P450s that are degrading the drug, maybe you can improve efficacy and reduce adverse side effect. This seems to be true, at least for blood pressure lowering drugs taken at evening time or bedtime, and much better in reducing long-term cardiovascular disease risk. And it seems to be also true for arthritis pain medications. Usually arthritis pain is more in the morning, but the pain medication with slow release taken at bedtime are much better. So we hope that this will spread to many other drugs. And then finally, drugging the clock. We now know that many cancers actually don't have a functional circadian clock. So then the idea is if we reactivate the clock in very pedestrian term, or specifically activate certain clock components, particularly the pressures, then we may be able to improve some conditions. And actually a few years ago in our lab, what we found is if we reactivate reverb by an agonist, then it would specifically kill oncogene-induced senescent cells or many types of cancers in a p53 independent manner. And we took it to glioblastoma in a patient derived glioblastoma transplanted into a mouse, and then we found that this candidate drug or the tool compound was much more potent than temozolomide in survival. Anyway, so I will change gear and talk about what I'm supposed to talk about. So almost 15 years ago, Joe Bass's lab did a very simple, but I guess this is most impactful experiment. And that is, he essentially put mice on regular chow for a few days and then moved them to high-fat diet. So RC is regular chow, high-fat diet is high-fat diet. And what you're looking at is the eating pattern or feeding pattern of mice and or activity pattern similar over 63 days. And what he found was on normal chow, regular chow, mice eat 15 to 18% of their food during daytime. And when you transport them to high-fat ad libitum, then these mice actually consume up to 30% of their food during daytime. For most people who work on nutrition, they would say this is a small change going from 15 to 30%. They're still eating mostly at nighttime. Majority of food is eaten during nighttime. But if we now combine this observation with our previous result that consolidated feeding increases circadian rhythm and circadian rhythm disruption leads to many disease, then now the question is, if we consolidate feeding in high-fat diet-induced obesity model, can we prevent or reverse some of the diseases that are linked to high-fat diet? So the experiment was very simple. We took high-fat, this was all done in male mice initially, high-fat ad libitum or high-fat time-restricted, and normal chow time-restricted or normal chow ad lib. These mice consume almost isocaloric diet for over 18 weeks. Their kilocal per mouse per day is almost equivalent among groups. The activity pattern actually changes slightly, but the total activity remains comparable between groups. And then the RER, as you would expect, the RER becomes more cycling under time-restricted feeding condition, both in high-fat and normal chow condition. And the result was surprising because the high-fat ad libitum fed mice, as expected, were obese, but then the isocaloric fed, time-restricted fed mice weighed 20+% less. The normal chow fed mice, between normal chow ad lib and time-restricted, there was no statistically significant difference in body weight. And this was kind of the picture of the mice. And so I showed you that they were eating isocaloric diet. And what we saw is the oxygen consumption slightly increased in both time-restricted feeding group and the postprandial state, not in the fasting state. So we think that there is slightly more energy expenditure in this mice. We don't know from where it is coming. Manuscript has many figures, 180 plus figure panels. So we looked at few things because it is obvious to see what the nutrient sensors CREB, mTOR, and NPK are changing, because by that time, these three were already linked to circadian clock components. What we found is CREB phosphorylation actually remains constantly high in high-fat or live-fed mice. Time-restricted feeding brings back this rhythm, and CREB should be phosphorylated in the light phase or during the fasting phase, and that's what we see. And the phospho S6 should be high only during the fed phase, but under high fat ad live, it kind of remains erratic throughout day and night, and time-restricted feeding brings back that rhythm. And we see AMP, at least AMP level kind of slightly goes up. It's not statistically significant. This is over 24 hours. And we also see improvement in many clock components cycling at mRNA level. Actually, the protein rhythms are much better now. And the takeaway message was time-restricted feeding, 8 hours time-restricted feeding of isocaloric diet, reduced body weight, adiposity, white adipose tissue inflammation, liver steatosis, glucose tolerance, glucose intolerance, fasting and postprandial insulin, plasma cholesterol, plasma triglyceride, and increase these factors. The normal chow-fed mice did not have too much difference. They had mostly statistically insignificant, some improvement, at least in 18 weeks. And then the question was, okay, this was a prevention model. You can prevent a lot of disease. For example, smoking cessation will significantly reduce the risk for lung cancer. But if you're a smoker and have lung cancer, quitting smoking after diagnosed with lung cancer doesn't help. So then the question is, can time-restricted feeding be therapeutic? So this is a big experiment that Amandine did with 500 plus mice. Essentially, he tried different types of diet also, high fat, high sucrose diet, high fructose diet, ad libitum or time restricted. And then to see whether just 5 days of time restricted, 2 days of ad lib, 2 days of cheating will help, because we know that for exercise, you can do exercise every single day or every other day, or even only for 2 or 3 days. It gives you benefits. So 5 days of ad lib, 5 days of time restricted, 2 days of ad lib. And then he wanted to fatten up the mice by giving them ad libitum and then, first ad libitum and then time restricted, and first time restricted and then ad libitum to see whether there is legacy effect. Then to see whether 8 hours is a magic number, because the reason why it was 8 hours was not empirically done. Christopher, who did the experiment first, he had a girlfriend who did not let him to be in the lab for more than 9 hours. So that's why he had to feed the mice for 8 hours. And to our satisfaction, mice actually eat isocaloric diet in 8 hours. So people always forget that there is a reason why. There's this weird reason for why we did this. So if Amandine does the experiment at 8 hours, 9 hours, 10, 12, 15, all of that she tried, then the bottom line is high fat, high sucrose. These are isocaloric intake. Again, high fat ad lib, 9 hours of time restricted feeding, 12 hours of time restricted feeding, 5 days of 9 hours, 2 days of ad lib. They're kind of eating similar calories, but then the 5T2A, these mice actually overeat. They consume more food than the ad lib fed mice. Then if we look at the body weight, the 9 hours and 12 hours have very similar body weight. 5 days of time restricted, 2 days of ad lib have statistically similar body weight. The fat levels also decrease. So this is the body composition, fat levels are comparable. 15 hours of time restricted feeding does not have too much benefit if we extend the experiment. So 12 hours seem to be, at least in mice, 12 hours seems to be the limit. And this is, I think, the most important experiment, where she fattens up the mice up to 25, 26 weeks. They are 55 plus grams. And then when you put them on time restricted feeding, they lose weight over the next 13 weeks. In long-term experiment, the high-fat time restricted fed mice are actually heavier than the normal chow. So the good thing is yes, diet quality does matter. On long term, the normal chow-fed mice are healthier or have less fat mass than the FTT mice. And there is no legacy effect. As soon as they go to ad libitum, they quickly gain back that weight. If we look at the normal chow-fed mice, what is a good surprise is time-restricted feeding actually increases muscle mass. So for mice at least, we don't know whether it happens in humans, male mice can increase their muscle mass as they get older, which is counterintuitive because we know that mice, as they get older, they lose muscle mass. But I guess at least for mice, we can preserve muscle mass. And then, of course, this paper also has 120 plus figure panels, but these are some interesting ones. So what Amandine found is just like in the previous experiment, whenever mice underwent any kind of time-restricted feeding, so anything that ends with a T, those mice were sacrificed when they were under time-restricted feeding. So there is less infiltration, macrophage infiltration in the white adipose tissue, and the takeaway message is brown fat is more brown under time-restricted feeding condition. If we look at the grip strength, then we don't see changes in grip strength in any of these groups. But if we put the mice on treadmill, what is interesting is normal chow-fed mice in this condition, the treadmill experiment can vary from one lab to another lab. In this condition, they run for 75 minutes. The high-fat ad lib, of course, they run less. All of these experiments, they involve mice that are equal body weight. So that's why the number of mice is less, because we are pre-selected. 9 hours of time-restricted feeding, these mice, they're eating high-fat diet, time-restricted. They outperform the normal chow ad lib fed mice by 100%. So they run for 150 minutes. And then the benefit actually goes away if they eat for 12 hours. These mice are equal body weight. They have similar muscle composition, but we don't understand why 12 hours actually reduces that. 5 days of time-restricted feeding for 9 hours preserves that endurance effect. And the same thing in other rotor rod performance, which monitors both motor coordination and to some extent endurance. There's a legacy effect. Any mouse that has ever gone through at least 10 weeks of time-restricted feeding, they can stay on rotating rod for longer time than mice that eat ad libitum. This is all done in male mice. So we wanted to do female mice and also young female, old female, young male, old males. And then we switched to 45% calories from fat, 17% from sucrose, this diet. And what we found is, again, both male and female, whether at 3 months or 12 months, they eat the isocaloric food. And as expected, actually male mice are more, as mice get older, they actually eat slightly more food during daytime than young mice. But under time-restricted feeding, as you would expect, we are giving all the food within 9 hours during nighttime. Although male mice are protected from weight gain under time-restricted feeding, at least in this experiment, we did not see female mice are protected from weight gain under time-restricted feeding, with the caveat that the female mice are actually weighing much less than the male mice. So we don't know whether if we continue this experiment for more than, say, 20, 30 weeks, we'll see any difference. But the bottom line is, at least in this condition, there is not much body weight difference between ad lib and time restricted in female mice. But at the same time, what we see is female mice do deposit more fat, triglyceride in their liver, and time restricted feeding protects them both at the young and older age. We also see, although time restricted feeding did not change body weight, it improved their glucose resistance in female and male mice. That postprandial and fasting insulin, actually the fasting insulin is the one that improves in both male and female. Of course, the female mice, we know they don't get as insulin resistant as the male mice are, but there is trend in improvement. And in older mice, we also see female mice, although they are not protected from weight gain, they do accumulate a good amount of liver fat, triglyceride, which is reduced by time restricted feeding. So this is it. And then finally, at the end of the experiment, we challenged the mice with the LD50 dose of LPS. And the LD50 dose for LPS for B6 male and female mice are slightly different. So we did the right dosing for the sex. To our surprise, what we found is female mice that went through time-restricted feeding are completely protected from LPS-induced death. And this is very important because in many caloric restriction studies, mice actually have compromised immune function. They can succumb to LPS-induced shock or infection much more than the ad-lib-fed normal calorie-fed mice in some, not all experiments. And in this case, even the male mice are also protected to some extent from LPS-induced death. So that was published a few years ago, and we are following up on that. And I guess there was a question from grad students saying whether we have done this in other species. And actually, we did this in flies. And in flies, we put them in normal chow or high-fat ad libitum or 12 hours time-restricted feeding when they are given access to food only during the daytime. At nighttime, they're transferred to simple agar so that they can still stay hydrated. And here we looked at actually the heart function. And some of you who may follow the fly heart literature, you can decapitate a fly, put the fly body under artificial hemolymph, open up the heart will continue to beat for 12 hours. So you have a long time to figure out how the heart is beating. You take 1 pixel width of this image and then plot them right next to each other, and you get an M-mode image, which looks, the grayscale image looks like that. From there, you can figure out diastolic, systolic diameter, heart period, arrhythmic index, radial contractility or fractional shortening. And all of these values move in this direction as humans and flies age. So it's a nice, simple model of looking at cardiac aging. And if we put these flies under time-restricted feeding, this is the ad-lib-fed fly. I showed you the video at 3 weeks. By 5 weeks of age, the fly's heart becomes very arrhythmic. And you can see that. And then the time-restricted fed flies, they are relatively more rhythmic. We actually quantify this, and we can go and quantify all those beats. And what we find is up to 3 weeks, 5 weeks, or 7 weeks, essentially the fly heart is aging 1 week slower than the ad libitum fed fly heart. This is found in both male and female flies, multiple different strains, and this phenotype is dependent on having a functional circadian clock because all the circadian mutant flies don't show the benefit of time-restricted feeding. And also this was dependent on mitochondrial function, particularly complex 1 and 2, as well as protein folding factor, particularly ATP-dependent protein folder, this CCT complex, and we can talk about it offline. So over the last several years, there have been many benefits of time-restricted feeding. So recently, we did a very comprehensive analysis of what is actually transcriptionally changing in multiple different tissues. So we harvested mice, took out 22 different tissues, every 2 hours over ad-lib or TRF, 1,035 samples. And we did 2 different kinds of analysis. One, differential gene expression that everybody can do, and then the other one was cycling gene expression. But first thing that we did is we made a UMAP to see which clusters, how many clusters we see are changing and where they're changing. The take-home message is, if you look at the E word, for example, this cluster gets down-regulated in TRF, and I word that gets up-regulated in TRF. So that means the same cluster of genes may have very different directional impact on the time-restricted feeding. So I'll not go into detail, I'll just talk about the broad numbers. So time-restricted feeding seems to have biggest effect on the adipose tissue. The next ones are the duodenum and jejunum, but not ileum. That was surprising. And then hypothalamus and adrenal, so that means the neuroendocrine system is also affected. So this is the pairwise, all these 20 plus, actually not 20 plus, because some of the brain regions don't show any differential gene expression. Whatever short differential gene expression, those are mapped here. And the bottom line is what we found, heat shock proteins seem to be upregulated in time-restricted feeding in multiple tissues. And then these are the leptin, and many of the inflammatory genes are downregulated. And those are the GO terms that are up or down regulated in 5 or more tissues. Then we can look at the rhythmic gene expression, and the hypothesis so far has been time-restricted feeding might improve circadian rhythm, but nobody had shown that. So we went in and asked what happens. Actually, in early fed mice, 40% of the genome is rhythmic in one or more tissues, and that number jumps to 65% of genes become rhythmic in different tissues. There are only two, SCN and another tissue, where the number of cycling genes actually don't increase, don't budge, but almost all other tissues show an increase in rhythmic transcriptome. Also, the phase of gene expression becomes more consolidated. So that means there is a big cluster of genes that peak toward the end of the fasting cycle, and in anticipation of feeding, they actually go down. And there was another cluster that are food induced. And if we look at the phase of the cycling genes in multiple tissues, that can be taken as a marker of synchrony or inter-tissue, inter-organ communications, then that synchrony also improves under time-restricted feeding. And if you look at which processes are improving or going down, and actually I'll just fast forward to this summary slide. So essentially, during the fasting phase, autophagy, DNA repair, cell cycle regulation, and fatty acid oxidation, these were upregulated. And in the feeding phase, it's mostly quality control of RNA and protein. Those pathways were upregulated. But if we dig deep and then say, one example, if we ask fatty acid uptake, release into circulation, then it's mobilization, reuptake in the liver, and release of VLDL particle, etc. All these annotations essentially suggested that maybe these mice may be less susceptible to atherogenic challenge. So that's why we took LDL receptor knockout mice. We know that almost all drugs now in clinical practice to lower cholesterol depend on LDL receptor directly or indirectly, whether it's PCSK9, statins, even ANGPTL, they all depend on LDL receptor. So these mice also respond to time-restricted feeding. This is a high cholesterol diet. They prevented from weight gain, and the serum triglyceride levels reduced by 20%, and we also see reduction in plaques by 30-35%. And this is very exciting for us because nearly 1 in 100 people do have familial hypercholesterolemia directly or indirectly linked to mutations in LDL receptor system, and they don't respond well to statin. So then the question is, can we combine statin with time-restricted feeding? This is again only mouse experiment, so I will not go into detail. So then the question is, let's see, we have only 4 or 5 minutes. So we asked, okay, so how much of this can be translated to humans? And the reason why I went to human studies was not by choice, but by I was sending all these applications to NIH, DOD, and I would get this question from reviewers saying that, well, this is interesting in mice, but humans don't eat like mice, so your research has no significance. I would get significance score 7, 8, 9. Innovation score 7, 8, 9, because they say, well, you are just changing the timing of food. So we wanted to see when people eat. And surprisingly, when I went back to many nutrition science major papers, no paper objectively monitored nutrition timing for more than 2 days. And we know that we eat differently between weekday and weekend. So we wanted to see when people eat. So we made an app. The first version of app was very simple. We took a clue from Amazon, one-click checkout. What you do in one click, you select what you want to buy and press one click, and then it's in your purchase box. So open the app, one click, take a picture, second click, press save, third click. Instead of saving the picture in the phone, the picture actually came to our server. So we asked these 156 adults who were not students, who were not shift workers, to take pictures of everything that they consume, even water. The only thing they have to do is take a picture. And so we got all kinds of pictures from 156 people. We got 26,000 images, and we arranged them in what we call fidograms. So essentially, if we decide that this image has 5 kilocal or more, so that means Diet Coke, black coffee, black tea, they didn't count. We did not count them, even though they might have added sugar. And this is the feedogram of one person. We asked them to monitor for 3 weeks. So that's why you have all this. And every time they ate, they have to log it. And if they log two things within 15 minutes of each other, that was counted as one event, because we also found out that when people are eating, say, 3-course or 4-course meal, then it takes 14 minutes to finish one person and then go to another person. So these are each meal. And if we combine this for the weekday and weekend, as you'd expect, we did see delay in breakfast time during the weekend. And if we combine this, and then this is 3 weeks of data for one person and put it around the clock, this is how it looks like. The top one is 6 a.m., bottom is 6 p.m. And you might find this is an outlier, but this is 156 people. This is how they ate. And then if we ask, you have to do some sanity check. The sanity check is, do you see a peak around noon and evening? Because that's when people eat lunch and dinner. So that's what we see. There is a nice peak at noon, nice peak in the evening. Since we have all the pictures, we actually put it in Amazon MTurk. That's the primitive intelligence. So essentially you ask the crowd to guesstimate how many calories are in each picture. So we have 3 or 4 MTurk. And what we discovered is many nutrition students are actually eager to guesstimate. And what we found was, as you would expect, this is estimated calories per picture, not meal, because a meal can have multiple pictures within 15 minutes of each other. As you would expect, the number of guesstimated calories in each picture is peaking around noon and evening, but then when people get up in the middle of the night to eat, they're not just stopping at half a banana or half a cookie. They actually were eating a bowl of cereal or tub of ice cream. So that's why the number of calories are pretty high during those midnight to 6 a.m. If we ask what is the 95 percentile chance, what is the time interval in which there is a 95% chance that this person is going to eat within 3 weeks interval, then what we find is 50% of adults have that window. That's 14 hours, 45 minutes, we call it. 50% of adults eat for 15 hours or longer. Less than 10% of people consistently eat within 12 hours or less. And so that's what we see. And then we asked, is it a modifiable behavior? Because people have gone through their whole life eating like this. If you tell them, can they stick to a short time interval? So this is where their baseline eating window is, and we ask them to self-select a time window they think they can stick to for 10 hours windows, and they have to do it for 16 weeks. And that's what they did. And we saw a modest 2.5% reduction in body weight. These people are not obese. They had overweight, average BMI of 27. They had a baseline eating window of 14 hours or longer. After 16 weeks, there was no contact with these people. They came back after 1 year, and we are surprised to see that even that modest weight loss was sustained. And when we asked them why, we were surprised that they said they slept better, because our mice, we did not measure sleeping. But what we find is in humans, the sleep satisfaction, it's not sleep duration or sleep fragmentation that's improving. And then they were feeling less hungry at bedtime, at least at 16 weeks, and they were feeling more energetic. So that was a positive feedback loop that was driving this. And then we asked, because in clinical setting, I don't know whether there are physicians here, or when people come with diabetes brings their own sinister friends. So people are not coming only with insulin resistance, they come with multiple different factors. So that's why we call it metabolic syndrome. That means they should have at least 3 out of these 5 different factors. And we asked whether people with metabolic syndrome can adopt time-restricted eating, because all those previous people, they are all healthy people. Because we know obesity and metabolic syndrome can also change your appetite, your hunger, satiety, all this score. And they're also on medication. So we asked them whether they can do, and 16 out of 19 were on different types of medications. And the bottom line is, yes, they could improve their health. And these are the benefits. We do not see increase in HDL cholesterol, but the non-HDL cholesterol did reduce. Orange is the eating pattern for 2 weeks at baseline. Blue is the eating pattern at the end of 12 weeks intervention. So you can see that no one is skipping breakfast. They're actually delaying their breakfast up to 2 to 3 hours after wake-up time, and they're advancing their dinner time so that they can finish their dinner at least 3 hours before their bedtime. And we had only 2 people with undiagnosed type 2 diabetes. You can say type 2 diabetes because their fasting blood glucose at morning was more than 125. So this is 2 weeks of CGM data, and that's the eating pattern. And you would think that this person is healthy because eating 3 square meals, 1 immediately after waking up, 1 around noon, and then 1 before 7 p.m. But the eating window was around 14 hours. The only thing that he did was to delay the breakfast. He brought breakfast to work and ate the breakfast at 9 o'clock. And that reduced blood glucose dramatically. So that's before and after. Why this is important? There are 90 million people in this country who have pre-diabetes, or they are undiagnosed early stage diabetes. When a pre-diabetic person goes to diabetes, the total healthcare cost per year goes up by $9,000. So that means if we can delay diabetes in 1 million people for 1 year, that's $9 billion in saving. That's much more than what NIH has spent on circadian rhythm in the last 50 years. At the same time, we thought, what about shift worker? Because whenever we talk about circadian rhythm, people say shift workers have the worst circadian rhythm. And we know that nearly 1 in 4 working adults in the US is a shift worker. 1 in 5, according to Department of Labor Statistics, but then they don't count food delivery workers and Uber drivers, Lyft drivers, all this. So it's better to say 1 in 4. Shift work disrupts circadian rhythms and increases the risk for all these diseases, but shift workers are almost always excluded from clinical trials because it's hard to schedule. And we also know that many drugs don't have that efficacy that much among shift workers. So the question is shift work. So then we asked, if you go to clinicaltrial.gov, 425,000 studies are registered and less than 400 studies are on shift workers. So then we asked, well, can time-restricted feeding work on shift work? And I'll actually not go into when we did this study, this was funded by Department of Health, Homeland Security. They asked us, how do you want to disseminate the results? So we had a videographer who went with 4 randomly selected shift work participants and followed them throughout the study. And we have a documentary that's actually going through now different film festivals, but this was the trailer. I'm not going to take time. But the bottom line is, this was a 1-year study, but I'll go talk about the first 3 months of results. We put a CGM on them. They had an active watch and they used the app. So you can see all the food that they ate. You can look at their glucose continuously, and their light exposure, activities, sleep, everything was recorded. And this was a 150 people study, but the union said you cannot select firefighters with metabolic disease because that would be a workplace discrimination. So it was first come, first served. So nearly half of the participants had normal health. And again, this is the standard of care group at baseline and post-intervention. The eating pattern was very similar. This is the time-restricted fed group. The red ones are the meals that they were eating outside their self-selected window at baseline, and they reduced that, not that they eliminated, they significantly reduced it. This is the cardiometabolic risk factor. So as I said, nearly 20% of them were almost healthy. Since it was a mixed population, you do not expect that they will improve their health. So they just reduced their eating interval significantly. The only improvement in the whole group was VLDL cholesterol particle size that improved in time-restricted feeding group. The weight reduced in time-restricted feeding group, but it was not significantly different between groups because you got to also think about between group analysis. And BMI also changed. Diastolic blood pressure changed within group, but not between group. So now, since it was a mixed population, if we take people or firefighters who actually had clinically significant high or worrisome level of these factors, and if we take, for example, fasting blood glucose, yes, time-restricted feeding actually improved their health significantly within group and between groups. Same thing goes for HOMA-IR, systolic blood pressure, diastolic blood pressure, LDL cholesterol. And so essentially all the metabolic risk factors, if they had one or more of those conditions, time restricted feeding reduced them. So I will stop there with the summary that circadian rhythm disruption by erratic lifestyle, whether it's light-dark disruption, sleep disruption, or eating pattern disruption, can increase the risk of many of this disease. And then circadian lifestyle, one of them is time restricted feeding, can prevent or reverse many of these conditions. Thank you.
Let's take one or two questions and then we'll leave the class. Anybody questions? General questions?
Dr. Taylor, thank you so much for your highly informative speech. And I'm just wondering, so in order to maintain the benefits of the time restricted feeding, that 8-hour window, do you think we should change that window, that 8 hours, according to the different chronotypes of, say, research subjects, patient, like in humans?
Yeah, actually what we do, we don't tell them to eat between 7 and 3 or whatever it is. So we ask them to select, self-select the 10 hours. We actually don't do 8-hour time restricted feeding because we know that the long-term compliance is low. We start them at 10 hours time restricted feeding, and it's a self-selected window, but we give them some clues. So for example, if you look at melatonin, over the last 15 years, one big discovery in melatonin field is one of the strong locus linked to obesity and type 2 diabetes is melatonin, one of the melatonin receptors, which is expressed in beta cells, and this receptor binds to melatonin much stronger than this allele binds to melatonin much stronger than the wild type, and that inhibits glucose-induced insulin release. So the implication is, for 50% of us, our melatonin levels begin to rise 2 hours before our habitual bedtime. And that means you should not be eating at least 3 hours before your habitual bedtime. So then people can self-select when they should stop. In the morning, most of us wake up with an alarm clock, and that means although our brain wakes up, our melatonin levels is still high and can remain high somewhere between 2 to 3 hours after wake-up time. So that means that's also a bad time to have your breakfast. So we ask them, well, wait for 1 to 2 hours after waking up to have your breakfast. So in that way, they self-select. So that's why in all of our study, we ask them as self-selected. So in that way, whatever you call chronotypes, they are self-selecting the window that works best for them.
Thank you so much.
Any more? I'll ask one question. So there are, in the popular press as well as the research press, intermittent fasting, caloric restriction, time restricted feeding. Do these different regimens, are they overlapping? Are they independent? What is your sense on that? I mean, from a mechanism perspective, would you expect additional benefits?
Yeah, so as I said, quality, quantity, and timing of food matters. And the mouse experiment also shows that normal chow-fed mice under time-restricted feeding are healthier than high-fat diet. Then if we ask caloric restriction, as you know, now it's very clear that most calorie restriction studies, almost all calorie restriction studies in mice also had a time restriction component. And Joe Takahashi's recent paper showed how to disentangle that. And the bottom line is, yes, some of the calorie restriction itself does have benefit independent of timing, but timing and calorie restriction can work together to increase benefits, at least for median lifespan. So now, if we think about, say, older adults, because all these age-related disease risk go up in older adults, 60 plus. At the same time, they may be sarcopenic obese, or they may be losing muscle. So then the question is, we know that calorie restriction is not an option for them because that will just make them worse with less muscle mass. So the big question is, how can we optimize nutrition quality, quantity, and timing? And we should not forget exercise, because to maintain that muscle mass, of course, our mice are actually moving around in the cage. They are not sedentary like. I guess that will be the next thing. Can we combine quality, quantity, timing, or at least start with timing, and then once people have some control over that timing, that they can combine optimum nutrition and exercise.
Okay.
Okay. Thank you.
Right, yeah, you should have you sign.
Yeah. Establishing a set feeding pattern. Food at the same time every day. Okay, so there was a question. Let me see.
That was just me. I was wondering, you almost
whether there is benefit to establishing a set feeding pattern. And I guess the answer is, we reanalyze the calorie 2 data. And we asked whether from the 5 days of food record, if we look at adherence to calorie restriction or weight loss, what we found is actually the timing window, the way we are calculating 95 percentile interval, that did not have much impact on weight loss. But again, this takes into account both the window and also day-to-day variation. So if we just look at the day-to-day variation in breakfast time, then people who maintained a consistent breakfast time, they seem to get another 4 to 6% extra benefit from calorie restriction.
Yeah.
But not the dinner time. It was the breakfast time.
That's interesting.
Which makes sense from circadian point of view, because breakfast is like the morning light that resets our metabolic clock. So that's what we feel. And then in calorie 2 trial, what we also find is the median timing window within which they ate was around 13 to 13 and a half hours. So that raises the question, like if we do calorie 3, can we train people to eat within say 10 hours consistently, and whether that will further improve the health outcome, at least keeping the breakfast time more reasonable, maybe within 1.5 hour interval, and also reducing the eating window to maybe 10 hours, because you know that many time restricted eating studies in the wild inadvertently reduces calories. So this might be another
to tease out
adherence to calorie restriction.
I was just thinking about it because what you told me about that pulse of melatonin and it declines, and so many of these feeding patterns have pulsatile nature of endocrine and response. And so that if they were synchronized and anticipatory, that, you could have like an entrainment effect and it would be more effective. I think we're creatures of habit.
So that's why, it's very nicely shown in mouse model that if you change the eating time by 4 hours in mice, then you do see disruption of metabolism and liver. And then when we think about all the breakfast skipping literature, it's essentially not skipping every day, but they're actually randomly skipping breakfast half of the day. And that might be causing this circadian disruption.
Yeah. Okay. It's fantastic talk. I absolutely loved it. I'm sorry I couldn't be there in person.
Thank you.
Great to see you. Bye.
Bye-bye.
Thanks.